Hi everyone, welcome to the INI CET Biochemistry May 2022 recall. Biochemistry topic was the most tough topic in INI CET May 2022. So this will be a session that you're all waiting for because so many questions from molecular biology techniques were there. So we will just see the questions. The first question is DNA protein interaction is studied using. So what are the techniques to study DNA protein interaction? The options here is DNA footprinting, DNA fingerprinting, ELISA, Northern blot. ELISA, we know that it is not for DNA protein interaction, so it is out. Northern blotting is for RNA. Then the confusion will come between DNA footprinting and DNA fingerprinting. So we will see what is it. DNA footprinting means that if we have a DNA like this, then you can see this DNA is bound to a protein. Okay, now this DNA is lysed using an enzyme called a DNase. So the DNase enzyme will lyse all the DNA except that region which is bound to the protein. So those region which is bound to the protein is taken out and it is studied using a DNA electrophoresis. Then that is what is called as, and find out which is that band, etc. So this is called as DNA footprinting. Why it is called footprinting? Because the footprints of the proteins are the one which is studied by using this technique. So then what is DNA fingerprinting? DNA fingerprinting means that we know that every individual, when compared to another individual, there are normal variations in the nucleotide sequence. They are called as the polymorphism. So because of this polymorphism, if a person's DNA is treated with restriction endonuclease, it will cut the DNA at a certain specific palindromic site so that it is producing a specific length, what you can say, the bands of the DNA bands are produced, DNA fragments are produced. So it is unique for that person. Okay, so this kind of fragments which we are getting as unique for a person, that is called as, if you are using restriction endonuclease, that is called as restriction map. And this restriction map can be used to fingerprint a person. For example, so what is shown here is, you can see here the DNA is extracted and it is treated with the restriction enzyme. So it will produce some bands and that is studied using DNA electrophoresis. So this is called a DNA fingerprint. So how are we going to apply this DNA fingerprint? So we can see here, this is the DNA fingerprint obtained from a suspected person from the scene of crime. So that we can see that this is producing certain unique bands. Now, from the scene of crime, we got this DNA sample and that is the DNA electrophoresis, DNA fingerprinting done to that particular sample. Now, we have three suspects here, suspect 1, 2 and 3. So, we have to compare the bands from the DNA that we obtained from the scene of crime to the suspect. So, the most you can see the suspect 2 is having the same band locus when it is compared to the DNA that we got from the scene of crime. So we can find out that suspect 2 is considered to be the person who has conducted or did the crime. Okay, so suspect 2 is proven to be this person who did the crime, and this technique is called as DNA fingerprinting. The same that we can use to find out the suspected paternity. So we have to find, there will be a band length that is produced from in the child, and we are doing a DNA fingerprinting of the mother, and we do the DNA fingerprinting of the suspected father. So if the band length is matching with the child, that is, if there is any band which is seen only in the father and in the child, it means that it is the person we are suspecting is the true or the biological father of that child. Okay, so this is called as DNA fingerprinting. That is, basically we are using a polymorphism as the principle to find out the DNA fingerprint of a person, and that is matched with what we are obtaining from the scene of crime. So this is called a DNA fingerprinting. Now this is another question. A patient with Ewing sarcoma presented with the EWSR1 gene mutation, and the technique shown in the image was performed. Upon fusion, a yellow fluorescence was seen. Identify the technique shown in the image and the finding. Okay, so this is actually a fish technique, and this is actually a fish that was done in interface. This is an interface fish. Sanger sequencing, definitely this is not Sanger sequencing because we are not doing it like this. Then IHC, this is not the technique of IHC also. Then what we have in an RT-PCR. RT-PCR will not be like this. So definitely the most of you might have answered this question right. So we'll just check what is this fusion and yellow fluorescence. So this is the fish technique. This is published in a journal, and this is a fish technique done in this case of Ewing sarcoma. In Ewing sarcoma, if there is this gene mutation, there is a production of a fusion protein. That is actually a fusion gene. From fusion gene, a fusion protein is produced. This fusion protein is actually joining of two exons of the two exon regions are joined, and it forms a fusion gene. So this can be studied using two techniques. Either we can use the fish technique, that is what is shown here. The same is what is shown in this image. That is what we can see is it's a fusion. Because of the fusion, see there are two regions in the there are two fluorescence that is used here. So in this fluorescence, one exon is having the color of red and another exon is having the color of green. Okay, now if there is a mutation because of the fusion of these two exons, what we get is a yellow color. So yellow color says that there is a fusion between two exons. This mutation is what is called as a EWSR1 gene mutation, where a fusion gene is produced, and that is what is seen. Upon fusion, a yellow fluorescence is seen. So this is the technique here is fish, and one more technique we can use is a RT-PCR. RT-PCR also we can use, but in RT-PCR, this is what the result that we get in RT-PCR. In RT-PCR, because of the fusion of these two exons, there is a particular band that is produced, and this band is having 342 kilobase pair. So if we can find a 342 kilobase pair band, then it means that the fusion, these two exon region has fused. So that is what we can see. This is the RT-PCR of a paraspinal soft tissue mass, and where we can see a band. This almost corresponds to the 342 kilobase pair. Okay, so if we get a fusion protein, then it will produce this band. You can see that band is not seen in any other tissue mass, but only in this case. So this is also seen in Ewing's sarcoma. So Ewing sarcoma we can study using two molecular biology technique. One is this RT-PCR. The second is the multicolor fish. So in multicolor fish, because of the fusion, we are getting a yellow fluorescence. So that is what is shown here, a yellow fluorescence, because the red and the green is mixing to produce that yellow fluorescence. Now this is a simple question, which is the inhibition depicted. So I have told you in my major videos that whenever we see something which is X, it is a competitive inhibition. So it is definitely it is a competitive inhibition. We just go through what are the other Lineweaver-Burke plot. So this is the competitive inhibition where it is X shape. Then the uncompetitive inhibition, which both lines go in the parallel direction, and V shaped. So V shaped is in non-competitive inhibition. Then in a patient with maple syrup urine disease, all the following amino acids should be restricted in diet except. So first we should know that MSUD is a disorder associated with the branched chain amino acid. So definitely we should restrict the intake of the branched chain amino acid in the diet. So we have to restrict valine, leucine and isoleucine. So all this has to be restricted. So the question is except. So the answer here is methionine. Methionine need not be restricted. Deamination of methyl cytosine form. This was also a very difficult question for you. So we'll just go through this purine and the pyrimidine. So purine, we know it is a heterocyclic two-ring structure, and in this is adenine is 6-aminopurine and guanine is 2-amino 6-oxopurine. And now question is about a pyrimidine. In the pyrimidine, what we can see is I'll start with uracil. So this is uracil. What we can see is an oxo here and oxo here. It is 2,4-dioxopyrimidine. That is uracil. Now what is cytosine? Cytosine is 2-oxo 4-aminopyrimidine. That is cytosine. Then what is thiamine? 2,4-dioxo, then what we can see is 5. This is 5-methyl pyrimidine. That is called a thiamine. So the question here is 5-methyl cytosine, which means in this particular case, what we can see is the 5-methyl, right? 5-methyl means there is a methyl group here. Okay, now this 5-methyl cytosine is deaminated. So let this be 5-methyl cytosine. It is deaminated, which means this amino group is removed, right? So when this amino group is removed, what we get is an oxo group will replace here. So what we get is 5-methyl 2,4-dioxopyrimidine. What is 5-methyl 2,4-dioxopyrimidine? It is thiamine. So what is the answer here? The answer is thiamine here. So all the following are true about Human Genome Project except. So this is a very confusing question. It took nearly 2 decades. Total gene is 20,500. Next generation sequencing is used. Took international collaboration. All the options seems to be correct. So when I searched, what I could find is that here I'll just tell you some of the highlights of the Human Genome Project. The Human Genome Project is a multinational consortium, multinational led project, like different nations or a project which was done with international collaboration from different nations. So that's why it's called a multinational consortium. It started around 1990 and it has ended. The first Human Genome Project is declared to be end in 2003. So it nearly took less than 20 decades. Then the Celera Genomics is a private agency which collaborated with the Human Genome Project, international collaboration that was actually a government-led project. So this was helped by a private project also. This is called Celera Genomics under the initiative of Craig Venter. And the idea of Human Genome Project came in the United States of America. So first human genome was mapped in the year 1996. In 1999, so the sequencing of the chromosome 22 completed, and the first draft of human genome in 2000, and the sequencing of the whole human genome, which include all the 23 chromosome, completed in 2003. So it took less than 20 years to complete the Human Genome Project. So now we'll see the results of Human Genome Project. The exons, the percentage is 1.1 to 1.5%. The number of protein coding genome, number of protein coding genes according to Human Genome Project is 30,000 to 40,000 genes. Average gene contains 3,000 base pair. The number of base pair in a gene average is 3,000 base pair. Number of proteins is 80,000 to 1 lakh proteins, and the human genome is consisting of 3.2 billion base pair. That is, haploid set is consisting of 3.2 billion base pair. These are some of the results of Human Genome Project. So now we'll go back. Took nearly two decades is a correct option. 20,500 genes. It is not correct. The result of Human Genome Project, when it is published in 2003, it is saying 30,000 to 40,000 genes. So this is not correct. Next generation sequencing is used. Definitely. Next generation sequencing is a massive parallel sequencing that is used if we are having more number of samples. Definitely next generation sequencing is used, and the technique is about a shotgun technique is used in the Human Genome Project and took international collaboration. It is also right. So what I feel as the answer is the total genes 20,500, because when the Human Genome Project was published, it is saying 30,000 to 40,000 genes, not 20,000 to 20,500 genes. Then banding technique used for dicentric chromosome. The G banding, NOR banding, C banding, R banding. So what are these banding techniques? G banding is something that we all know, that is GMSA banding, where we can usually see as a light and dark pattern. Then quinacridine banding is otherwise Q banding. Quinacridine banding is similar to GMSA, but the examination of the chromosome is using a UV fluorescence microscope. Then reverse banding. Reverse banding is the chromosomes are denatured. That is a difference here. Here the chromosomes are denatured, and hence the light and dark pattern is reversed. What we get in light in GMSA will be dark in reverse banding, and hence the name R banding. Then what is C banding, otherwise called a centromeric banding? Centromeric banding is specifically used for heterochromatin regions. So heterochromatin regions are specifically stained, that is called as C banding. So now we'll see the banding used for a dicentric. So dicentric should be a banding technique that we use specifically for heterochromatin regions, which is a centromere. So the answer here is centromeric banding. So the answer is C banding. Now the next question is in Sanger sequencing, dideoxynucleotides are used because dideoxynucleotides are used because it binds and breaks so that DNA polymerase can't act further. That is the answer here. Which is used to increase the DNA polymerase? No. It is fluorescent? No. DNTPs are used because it exhibit higher affinity for DNA polymerase as compared to DNTP. No, it is also not correct. So what is it? So first we'll see the principle of this, that is in deoxynucleotide, when it is compared to dideoxy, what is the difference? The difference is in the ribose, in the sugar. That is what we can see is normally in deoxynucleotide there is a free 3' hydroxyl group. This is required for the formation of 3' to 5' phosphodiester bond. Now in dideoxynucleotide, in the 3' position, there is no 3' hydroxyl group. It is only a hydrogen, which means if a dideoxynucleotide is incorporated, then further chain elongation will not occur, which means there is a chain termination, because there is no free 3' hydroxyl group to form the 3' to 5' phosphodiester bond. This is the principle of Sanger sequencing. So if we have a sequence like this, that we are going to sequence, if we have an oligonucleotide like this, suppose we are using a dideoxy A. Now to the A we know that T will bind. It is a normal deoxynucleotide that is binding. Now here what we can see is in the second set the A is binding. This A is actually a dideoxy A. So when the dideoxy A bind, what happens further? Actually the C has to bind with the G. But if the G is coming here, it needs a 3' hydroxyl group here. But that is not there, which means further chain extension will not take place, which means we'll get a band of 2 base pair length here. Similarly, if we are using a dideoxy G, what will happen to the C? Instead of G, sometimes G can bind, sometimes dideoxy G can bind. So if dideoxy G bind, then what will happen? Then it is the chain length cannot elongate further. The chain cannot elongate further, so we will get a 3 base pair. Likewise, these dideoxynucleotides is used for controlled chain termination, and because of this controlled chain termination, the chain elongation will stop whenever the dideoxynucleotides are added, and from the information, like if it is getting a each of this dideoxynucleotide will be having a particular fluorescence, and based on that we can find out what is the sequence that is present in the DNA that we have added for sequencing. So if you want to know more about it, you can just see my video of DNA sequencing, where I have clearly explained how the DNA sequencing is being done. Now this is also a very tough question. Correct sequence of steps in isolation of desired DNA fragment in recombinant DNA technology. Here the sad part is which technique they are meaning is not clearly mentioned in the question. So I will just give you a technique where we are using to isolate a DNA fragment after recombinant DNA technology. So this is actually a colony of, this is about a cDNA, like recombinant DNA technology that we are using a cDNA. So this is the colony that we obtain. So what we'll do is we'll first isolate the DNA from the clone. That is the first thing that we use. So we will do a bacterial cell lysis, and then we isolate that DNA out. So that is the first thing. Then what we'll do is so first bacterial cell lysis, then isolate the DNA. Then the DNA is denatured. So what we can see, the DNA is denatured. Then it is blotted to a nitrocellulose membrane. That is the blot. Then once it is blotted, in this particular technique, as it is a cDNA, we'll add some mRNA. Now mRNA will bind, so these are the cDNA, the mRNA will bind to the cDNA, and unbound mRNA is removed, and bound, that is mRNA, cDNA, bound DNA fragments are eluted. So the next step is elution of the, so mRNA is added, and what happens there is hybridization. Okay, unbound mRNA is removed, and these hybridized mRNA cDNA is eluted. So that is called elution, and whatever we are getting from this elution, we are doing a electrophoresis, like SDS page or any DNA electrophoresis technique that we are doing in this particular case. So we will just see the question now. The correct sequence of steps of isolation of the desired DNA fragment. So first in the set of list that is given here, what I feel the first should be the desired gene integration into the bacterium. That is recombinant DNA technology. We are doing that. So this will be the first step. Then the second step is the bacterial cell lysis. Then once the cell is lysed, then we have to isolate the gene of interest. So that is we are isolating the gene of interest. Then the third step is once the gene is isolated, we have to denature it. That is the fourth step. Then we add a specific, depending upon what is, if it is a DNA, we will add a DNA which is complementary to it. If it is a cDNA, we will add an mRNA, like that. Whatever is the situation, we will add a probe or a complementary sequence, or if it's a cDNA, we will add mRNA, or if it's like that. So we will add a probe, complementary DNA, and what is when we add the complementary DNA, what will happen? The complementary DNA will bind to the desired DNA fragment. So that is the fifth step, hybridization. So depending upon what is it, if it's cDNA, we'll add mRNA. If it's a DNA, we'll add a complementary DNA. So hybridization, and then the hybridized DNA should be eluted. So that is the next step. elution of the hybridized DNA. Then once that is done, we are separating whatever we have isolated. That is the last step. Seventh step. This is the sequence. This is a general sequence of steps of isolation of a desired DNA fragment in recombinant DNA technology. Now we'll go to the next question. Misincorporation of uracil in the DNA is seen in the deficiency of. So it is seen in the deficiency of B12. Folic acid is also right, but more correct I feel is the B12. Because this is a diagram that is given in Robbins. Here we can see in the step of methionine synthesis, where methionine is converted to, homocysteine is converted to methionine. We know that the methyl B12 folic acid is the methyl donor. It becomes the free THFA, and this is received by the B12 and it becomes methyl B12, and methyl B12 is the one which is giving methyl group to the homocysteine so that methionine is formed. So by this reaction what we are getting is we are actually freeing the tetrahydrofolic acid. So if there is a B12 deficiency, what will happen? If there is a B12 deficiency, this conversion of methyl tetrahydrofolic acid to tetrahydrofolic acid will not happen. So there is a deficiency of tetrahydrofolic acid. If there is a deficiency of tetrahydrofolic acid, there is a deficiency of N5, N10 methylene THFA, because for that formation we need tetrahydrofolate, which is not there, and that is required for the DNA synthesis at the step thymidylate synthetase, which is actually a synthesis of pyrimidine. Now for the pyrimidine synthesis, this tetrahydrofolic acid in the methylene form is required, which is not there, which means the uracil is not able to get converted to TMP. So this will affect the DNA synthesis. This is the reason for megaloblastic anemia. Okay, so the question is misincorporation of uracil. So uracil is not actually converted to the TMP, and incorporation of the DNA synthesis is affected. So basically what is the vitamin that is deficient here is B12, because B12 deficiency is the one which led to the folic acid deficiency. So the most preferred answer here is B12, but pure folic acid deficiency can also cause this, but in the case of specifically in this case what I feel is the B12 deficiency more accurate than a folic acid deficiency.